there are two types of ribosomes. One is 80S ribosome and second one is 70S ribosome. 80S ribosome are majorly responsible for all the protein and peptide synthesis. 80S ribosomes are present in cytosol, whereas 70S ribosomes are present in organelles like mitochondria and chloroplast. Okay, so all the proteins are made by these ribosomes. We will be considering most of the time the 80S ribosomes, and the proteins made by 80S ribosomes may be needed by other organelles or other locations inside the cell. For example, all the proteins that are made by these 80S ribosomes, some of these proteins may be required by plasma membrane. Some of these proteins may be required by endoplasmic reticulum. Golgi may need some of these proteins, maybe lysosome, even cytosol. So many of the organelles has different requirements for the proteins, and each and every protein has to be synthesized by a ribosome, and that too majorly by 80S ribosome. So these proteins will be synthesized inside the cytosol, and they have to be transported to these organelles like endoplasmic reticulum or Golgi. And this mechanism, this process of transport of this protein to their native locations is what we call protein targeting. So protein targeting is the process by which newly synthesized proteins are directed to their correct cellular location. This is protein targeting. There are two terms that are often confusing. One is protein targeting, another one is protein sorting. So protein targeting begins very early. It is a very early step which begins when the protein gets synthesized by the ribosome. Whereas in case of protein sorting, let's consider that we have endoplasmic reticulum, and I am taking out one tube of endoplasmic reticulum. We have freshly synthesized protein from a ribosome attached here. This ribosome has inserted this protein, and this insertion is a part of the protein targeting itself. So we have different proteins like this, such as protein. So I have shown three proteins: protein number 1, protein number 2, and protein number 3. Suppose protein number 2 is required outside the cell. Maybe it is some kind of peptide, signaling peptide, which is excreted out of the cell. Okay, and for example protein 3 is required by lysosome. So these proteins, all three of them, will be distributed specifically to be targeted to different locations like secretory protein, like lysosome. This distribution of proteins is known as protein sorting. So protein sorting occurs later on when these proteins are acquired by an intermediate organelle like endoplasmic reticulum. Okay, so there must be some signals used for protein targeting or protein sorting. For protein targeting, different signals are used. Such signals are usually called localization signals or some specific targeting signals, whereas in case of sorting, we have sorting motifs which are required for sorting of these proteins. So we have two different type of targeting systems in protein targeting. One is endomembranous system, another one is non-endomembranous system. Remember that in case of endomembranous system, protein is synthesized and inserted into endoplasmic reticulum. From endoplasmic reticulum, the protein is transported to Golgi apparatus. From Golgi apparatus, through vesicular transport, it can be transported into various vesicles or lysosomes, or it can be targeted to plasma membrane, or it can be secreted outside the cell. And the proteins which are targeted this way are like insulin, which is required by different cells. There may be immunoglobulins, your antibodies. In case we have non-endomembranous pathway, non-endomembranous system. For this we have specific organelles like mitochondria, like chloroplast, like peroxisomes, like nucleus. And the protein transport to these organelles is very specific. For example, citrate synthase is a transporter to mitochondria. Several transcription factors are transported to nucleus. These pathways are summarized in this diagram. You can see the ATS ribosomes are synthesizing the proteins, and these proteins can go by endomembranous pathway, where they are inserted in a rough endoplasmic reticulum. They are targeted to Golgi complex. From Golgi complex, they are either transported to lysosome, or they can be inserted into plasma membrane, or they can be secreted outside the cell. Whereas the same proteins, or the specific proteins, on the basis of their specific sequence, can be transported via non-endomembranous system, where if they lack any transportation signal, they can be retained in cytosol. If they have specific targeting sequence, for example, they have a nuclear localization signal. If they have NLS, nuclear localization signal, they are transported to nucleus. If they have a specific sequence for peroxisome, they are transported to peroxisome. If they have specific sequence for mitochondrion, they can be transported to mitochondrion. If they have sequence for chloroplast, they are transported to chloroplast. So, in this diagram, on the left side we have endomembranous system, on the right side we have non-endomembranous system. Let's continue. What is the fate of protein? The proteins are synthesized by 80S ribosomes or 70S ribosomes. If they are the proteins synthesized by 80S ribosomes, they can enter cytosol and can be transported to different locations from the cytosol, or they can be retained in the cytosol. Now, the question is, what kind of proteins should be retained in the cytosol? As we talked about earlier, the proteins which lack any specific targeting sequence are retained in the cytosol. What kind of proteins do we need in the cytosol? We need hexokinase, we need some cytoskeletal proteins in the cytosol, we need ribosomal proteins in the cytosol. These proteins can be targeted and enter in various organelles like nucleus, like mitochondria, chloroplast, peroxisome, or endoplasmic reticulum. Endoplasmic reticulum is a specific case. In case of endoplasmic reticulum, transport can occur in two different ways. It can occur co-transcriptionally, which means that ribosome attach to endoplasmic reticulum and protein is synthesized and transferred in the process of synthesis itself. Whereas in another mechanism, it can be transported post-transcriptionally. Another fate is if it enters secretory pathway. In secretory pathway, there are secretory vesicles involved. It can be transported to lysosome, it can be transported to plasma membrane, from ER to Golgi transportation in vesicles, and it can be exported outside as well, like we see in insulin transport. Okay. And 70S ribosomes are responsible for synthesis of some proteins in the specific organelles like mitochondria and chloroplast. But the point to remember here is the signaling or the targeting in case of mitochondrion and chloroplast is internal and limited. Okay, so if there is internal targeting in mitochondria, the targeting will be between the mitochondrial membranes and matrix. Same with the chloroplast, the targeting may be to the stroma, maybe to thylakoid membrane, or simply lumen. And if it stays like it stays freely, then it will stay in the matrix of mitochondria, in stroma of chloroplast. Now we will talk about the type of transport. There are three major modes of transport. First one is transmembrane transport, second is gated transport, and third one is vesicular transport. Okay, and we will be considering these transport mechanisms from proteins synthesized via ATS ribosomes in the cytosol. Let's start with gated transport first. Okay, the gated transport is shown in a red color. The cytosol to nucleus transport is gated transport, and sometimes this transport, the peroxisomal transport, is also considered as gated transport. Alright, so gated transport has to occur through a selective pore or channel complex. For nucleus, we have nuclear pores. So it requires a selective pore complex This is bidirectional transport. Proteins which are transported through this mode are usually folded proteins. In case of transmembrane transport, the proteins transported are in unfolded conformation. Usually the proteins are unfolded. And transport of proteins occurs through different compartments. For example, we are transporting protein from cytosol to plastid. This is transmembrane transport. We have the transport to mitochondria. We have transport to endoplasmic reticulum. All this is transmembrane transport. And the third one, vesicular transport. In case of vesicular transport, we have two organelles. This is donor, this is recipient. A vesicle will be formed, vesicle will ward off, and the protein to be transported will be inside this vesicle. Vesicle forms, vesicle bud offs, and vesicle will fuse to recipient, and protein will be delivered. The soluble proteins and membrane bound proteins both will be transported by this mechanism, and the orientation will be conserved. Here you can see these are the membrane bound proteins, which are integral membrane proteins. Look at the conformation of these proteins. Look at this spherical domain, which is on the cytosolic phase, and this pointer domain, which is on non-cytosolic or lumen phase. Okay, so this is the donor compartment. The soluble proteins are packed in this Wadding vesicle. Some of the membrane proteins are also packed in this Wadding vesicle. The wadding of this vesicle takes place, and this vesicle fuses to recipient compartment, and you can see the soluble proteins are being transported to this recipient compartment, and due to fusion of this membrane, these membrane proteins are also transported. Now again, look at the orientation of these membrane proteins. The spherical domain is towards cytosolic phase, whereas the pointing domain is towards the luminal phase or towards non-cytosolic phase, and this orientations remains the same in donor as well as the recipient organelle. This was vesicular transport, and this orientation is conserved, or this directionality is ensured by vesicle coating proteins like COP1, COP2. We will talk about these proteins in details in the ER and Golgi or vesicular transport mechanisms separately in different video. The proteins synthesized by ATS ribosomes can enter transmembrane pathway, from which they can enter secretory pathway, go through endoplasmic reticulum, go through Golgi apparatus. They may be targeted to lysosome, they may be targeted to plasma membrane like we saw earlier. They can be transported or exported outside the cell, just like in insulin. In case of gated transport, proteins like transcription factors may enter nucleus, and some proteins which lack all these signals may be retained in the cytosol, or even cytosolic proteins can enter vesicles and enter the vesicular transport pathway. For the 70S ribosomes, if they are retaining freely, they may be retaining in the matrix or stroma of these plastids or mitochondria, and if they are membrane bound, they will be localized internally. They will be targeted internally between the membranes of mitochondria itself, or if it is chloroplast, it will be retained in the membranes of chloroplast itself. Moving on, what is the mechanism of transport? We will talk about the general mechanism of transport of this protein targeting. The protein which is synthesized by ribosome, it may or may not have specific sequence of amino acids known as targeting signal. On the recipient organelle or intermediate organelle, we may have receptor for targeting sequence. Okay, so this targeting signal is recognized by receptor which is present on recipient organelle, and if this recognition is positive, the translocation channel will become active and it will allow the entry of this specific protein, and this entry is governed by some kind of energy source. This energy source is mostly in the form of GTP or ATP. This is the general mechanism I talked about earlier, that there is some sort of signal on the protein synthesized by ribosome which is recognized by receptor. Okay, and there will be some sort of delivery mechanism. and this delivery mechanism will activate some kind of translocon or some transporter protein which is governed by ATP or GTP. After this process, the signal is either retained or removed. Mostly it is removed, and the maturation takes place. By maturation we mean if the protein transported is in unfolded state, it must be folded with the help of chaperones, and some other kind of modifications may take place like glycosylation or another post translational modifications can occur. So the protein synthesized can have a sorting signal sequence. If there is no sorting or targeting sequence, the protein will be retained in the cytosol itself. And if it is having the specific sorting or targeting sequence, the nature of sequence will determine where this protein will go. Protein can be having a linear signal sequence or it can be having a patch sequence. If the sequence is linear, the sequence may be terminal or internal. For example, we have this peptide, we have N terminus, we have C terminus. The sequence may be at C terminus, the sequence may be at N terminus. Most oftenly the sequence is at N terminus. Alright, and the sequence may also be internal. If the sequence is internal, it is non-cleavable. If the sequence is signal patch, it is non-cleavable, it will retain. And if the sequence is terminal, it can be N terminus or C terminus. If the sequence is at C terminus, it is again non-cleavable, and if it is on the N terminus, it may be cleavable or non-cleavable, both. Mostly at N terminus it is cleavable sequence. So these signal sequences are usually 15 to 60 amino acid residues long, and they act as address for the proteins to be delivered to their respected organelles. Okay, so mostly these can be found in N terminus, and often they are cleavable as I talked about earlier. If they are internal, they are non-cleavable, and they are recognized by their sorting receptors present on the target organelles itself. Let's talk about some organelles and specific signal sequence features with respect to those organelles. In case of endoplasmic reticulum, hydrophobic amino acid sequence at N terminus, and which is recognized by signal recognition particle receptor. In case of mitochondria, we have amphipathic sequence at N terminus, and it is recognized by TOM receptors, which is translocase of outer mitochondrial membrane. In case of chloroplast, we also have N-terminus sequence, which is rich in serine and threonine amino acids. Okay, and it is again recognized by translocase of outer chloroplast TOC complex receptors on the chloroplast. For the nuclear localization, we have nuclear localization signal, most oftenly known by the name NLS. It is rich in lysine and arginine sequences, and such kind of motif can be seen in such signaling sequences. Okay, for peroxisome we have PTS1 or PTS2 complexes. PTS1 is at C terminal, whereas PTS2 is at N terminus. PTS3 is made up of 3 amino acids, which is serine, lysine and leucine, whereas PTS2 is a nonapeptide which is made up of 9 amino acid residues. In case of lysosomes, we have mannose 6-phosphate modification, which is recognized by mannose 6-phosphate receptor on lysosomes. Let's talk about sorting receptors. They specifically recognize the targeting sequence. Their functions is recognition, targeting, transfer and recycling. For endoplasmic reticulum, we already talked about SRP sequence, and it is recognized by SRP receptor. For the mitochondria, we have TOM complexes. For inner membrane, we have TIM complexes. For chloroplast, we have TOC complexes. Nucleus, we have alpha beta importins and exportins. For peroxisomes, I talked about C terminus PTS1 and N terminus PTS2 lysosomes. We talked about mannose 6-phosphate modification and mannose 6-phosphate receptor. Here are some specific signal sequences with respect to nuclear import, export, import to mitochondria, plastids, peroxisomes, endoplasmic reticulum, and some retention sequences.